Mr. Trouble. His first name is Winifred. They call him In for short. 65-year-old guy. He shows up for a discussion. Gleason 9 gets an implant and EBRT and 18 months of ADT. 10 months post his RT, he's castrate. He had nadered to 3-1, but now he's got biochemical progression, standard imaging without evidence of disease. He gets apalutamide, has a response, brief, and then progresses. Gets a flucyclovine PET eventually, and shows 4 to 5 bone mets and small pelvic nodes. He gets docetaxel times 6, but now he's got biochemical progression and he's symptomatic, evidence of disease progression. So if you had such an agent, okay, for those of us old, we used to call this difungal muctate, but now it's got to be a monoclonal. So it targets a cytokine or chemokine, which prevents dissociation of G-coupled proteins, doesn't appear to have AR activity, and you do a phase 1-2 study at the end of the line, and you see some activity, a little bit of toxicity, fatigue, but really pretty well tolerated. So how are you going to do the study to get this drug to move forward? So this is even an older version of Howard Scher's clinical states model, the original version published in Urology. Obviously, 18 years ago, this concept was not really attractive to get published in a sort of a higher profile journal. And the reality is that life's gotten much more complicated, as we all know. The timeline, eventually the font is going to have to get really small to keep sticking things in here. It was a little challenging to get the apalutamide in. All right, so as we've seen from Charted, this publication led to practice change in the U.S., followed shortly thereafter when Stampede convinced the Europeans that this wasn't just an American effort to use docetaxel earlier. We're now seeing patients routinely in clinical practice that got docetaxel in this setting. Many of us have treated patients with docetaxel, and I think it's most folks' clinical appreciation that for the most part, docetaxel appears to have activity in this setting. What we don't have is the follow-up on the changes that have occurred subsequently. So latitude, right? Those of us who have introduced this into our practices as a real-time phenomena, so we don't really truly understand what's going to happen to these patients, although we can imagine what's going to happen as we think about using a AR-directed paradigm for MCRPC. And then, of course, the most recent data that we've seen. So we're right now in a paradigm that we can argue about the utilization of AR-targeted agents of whatever stripe in a non-metastatic setting. It's unequivocally it's happening already. It's going to happen to a greater extent. Whether or not these patients undergo next-generation imaging and really have low-volume mets or not, et cetera, there's going to be an upswell of the use of these therapies. So as the entire paradigm moves leftward, we're left with Mr. Trouble, and we're going to see Mr. Trouble in clinic. The likelihood that he is going to respond to another AR therapy is unlikely. This patient may or may not have received docetaxel in a relatively short period of time, and perhaps the use of therapies even earlier may be problematic. But if you're going to use a cytotoxic in that setting, meaning a non-AR directed therapy for a patient who may not be a radium candidate, if you're in a community practice, you're basically at the end of the line. You've already now used up your entire therapeutic armamentarium, and you're sitting there trying to figure out what to do. And at the same time, that's the same paradigm that we're going to be pulling our patients to think about new drug development clinical trials, right? So that's the issue before us. I don't have any answers. So one of the things that is going to be left with is that as we've used these therapies, what do you think about how we're going to do drug development? So if you have a biomarker-directed therapeutic, you're golden, right? So you do a biomarker-directed clinical trial. Look at the genomic trials, DNA mismatch repair, you can identify, you can do a trial, the FDA loves it, you can move forward. But if you don't have a biomarker-driven trial, are you going to go to the end of the line and do trials after patients have had basically every therapy, try to repeat the satraplatin experience in a different era, hoping that you can still demonstrate a survival benefit? Those are the challenges that we're faced with. So the compound that we're talking about up there, which may not have a biomarker to guide it, is in the same boat in many ways as some of the immunomodulatory trials that are ongoing, the multiplicity of trials of checkpoint plus multiple agents, which are rational designs. Whether or not any of those will be so paradigm shifting that you can then enable those studies to proceed, I think is the challenge we face. So at the end of the day, those of us who obviously take care of these patients and do clinical research are going to be faced with this very rapidly changing landscape. And if a pharma company is trying to do drug development, to try to fully appreciate the implications of these changes in order to design a trial and pick a space to do it, I think is going to become increasingly challenging. It's good that the FDA in some ways is becoming a little bit more teddy bearish-like in terms of discussions, looking at novel endpoints that they had previously not accepted. I think there's going to need to be an increasing partnership there and a little bit more of an open-minded approach to drug development, or we're going to stall for a while. With that, I will stop. Dean did say that, and it's still true.
Rob, the use of the newer antiandrogen inhibitors do seem, in my mind, to have created some opportunity, because now, once you go through the ADT and you're going through Abienza and you're coming out on the other side, you know, everything we do from then on is pretty limited in effectiveness. And yes, we do get occasionally some meaningful taxane responses. Occasionally, you know, radium's hard to evaluate, as you know. CYP-T, I don't think we're going to talk a lot about. And so I'm kind of seeing a window open rather than a window closed, because the new therapies are all going up front. We're using them, and then all of a sudden we have nothing to do.
I agree, if you had them, and I think the talk we just heard elegantly points out that, boy, it's really hard to develop drugs in AR resistance, right? I mean, a lot of the things that we were pretty excited about have sort of tanked. So, but no question, I mean, if you develop a way to overcome resistance and have a drug, you have an open space. No question.
So I actually, I think we're going to get beyond the era of saying abiraterone enzalutamide resistance as if that's one entity, because we all know it's not. And we now have a series of studies that are looking at DNA repair. So now we're carving out that niche. We've got the very small MSI high niche, but we also, 50% of our patients in CRPC are P53 mutant, and we have these biomarkers of badness, but we're not able to target the biology of them. And so I think we really need to think about how we can maybe develop a drug in abiraterone enzalutamide resistant P53 mutant patients or something like that. I was thinking about this yesterday. I was putting the renal cell talk together. Like, look at VHL wild type versus VHL mutant. They have a totally different clinical picture, and yet we kind of lump them together.
Well, Chuck, you're absolutely right.
Yeah.
Look, there's no question that the ability to develop a biomarker-directed therapeutic intent trial is the way we're going to have to go forward. It's not the same disease. We all understand that.
But there's a biomarker, and then there's a therapeutic target. And like with P53, they're not the same, right?
Things that are druggable and not druggable, right? There are things that we used to think couldn't be drugged, can be drugged. So that may develop.
Well, I've been involved with radiopharmaceuticals for a long time, and one of the reasons I like it is you get away from the molecular heterogeneity, which in turn is undruggable the majority of time. I mean, that's just sort of a fact.
And that's where like lutetium will come
Absolutely.
into play.
No, the PSMA lutetium, I'll be talking about that a little bit later today, but it is a little bit exciting because I don't really care if it's P53 mutant or not. If you get the radiation in the right spot, boom, you have a solution. At least that's the theory.
No, but
the practice,
that's exactly how drug development is going to have to go forward, right? I mean, but it's clearly going. I mean, we thought it was complicated before.
Yeah. And
it's not just that there's so many drugs. It's just prostate cancer, because of the unique things that we know drive this disease, make it even more challenging to do drug development.
Yeah. I like the tempered enthusiasm. A little bit exciting.
Yep. All right.